can't think of any part of the brain as existing in a dish. But the idea that you would place the pressure, the computational pressure of an automatic function on something as capacity limited as the basal ganglia never really meshed with me.
Welcome to Stimulating Brains.
Hi, it's Andy. Welcome back to Stimulating Brains, episode number 9. When I started the podcast project, I thought of it as a very broad concept to interview people, of course, in the field of deep brain stimulation, but also anatomists, physiologists, people that try to figure out how the brain works, people that have radical new ideas of how deep brain stimulation might exert its effects on the brain, people that study non-invasive brain stimulation, people that are looking for better ways to map anatomy of the brain. So basically, to interview stimulating brains that are able to move the field forward. So far, I've mostly interviewed people that were clinicians and important in, especially I think, the history of deep brain stimulation. I've interviewed Lone Frank as an author, but she had written about Robert Heath, so again, a historical figure of DBS. And recently there was the excellent guest episode by Luka Milosevic with Mojgan Hadai. That was a pure joy to listen to for myself as well. So hopefully there will be more guest episodes, and hopefully we will interview as many clinicians and experts in DBS as we can. Still, I think the scope and the idea of the podcast is broader than that. And today's episode may be the first one where we dive further into how the brain works, of course, with a focus or some context of the brain stimulation or the basal ganglia, or in this case, even the basal ganglia, thalamus, cerebellum, and cortex. I talked with James McShine, who is a brilliant scientist in Sydney, Australia, and who recently wrote, in my view, a phenomenal paper that comes up with a radically new theory of how these aforementioned systems may interact. It extends or partly even confronts existing theories, such as that the thalamus is a mere relay station in the brain, or even more importantly, also the Alexander DeLong and Strick model from 1989. His paper is quite dense, but we still try to focus on the paper and really go into depth. And I think and hope that we were able to get across the main points of the paper, still with the notion of motivating every one of us to delve into that paper and even more so into the underlying literature. So Mac invites us into a long journey of how the brain may truly be wired up, and to think about these ways and concepts and their implications. I hope you'll all enjoy this particular episode, and I'm, as always, curious to hear about feedback of what you think. Thank you so much for tuning in, and now have fun with James McShine from Sydney. So Mac, thanks so much for taking part in this. You were just mentioned a week ago by Russ Poldrack in an episode of the brilliant Brain Inspired podcast by Paul Middlebrooks, and I think Russ mentioned you sucked him into the field of dynamic network theory, screaming and shouting back when you were in his lab at Stanford. Why do you love attractor state so much?
Yeah, that was quite a spin out. I think I've listened to every single episode of Paul's podcast, and I absolutely love it. And then to hear Russ mention my name on the podcast, I think I was blushing as I was walking around listening to the talk. So attractors are a really, really exciting, interesting idea that make a lot of really complicated mathematics much more intuitive. And so I've gone on a really long and winding journey through a lot of different subfields that I see as being very important for sort of really understanding the brain at the end of the day. things like fields like cognitive neuroscience, fields like neuroanatomy, fields like clinical neuroscience. But one of the more challenging conceptually is the field of complex systems and dynamical systems, because the language there is really written in mathematics, which is not my strong suit. And so for me to understand what collaborators are thinking about, or what are really great papers are writing about, for me, I really needed intuition pumps. And I think attractors and attractor landscapes and all the things that go with them offer that for the non-specialist, the non-physicist, the non-dynamical systems expert. And so I've really tried to kind of capitalize on that intuition and use that to help me really understand what I'm reading about, hoping that also someone that comes along that reads it will have the same background maybe as I do, and say, oh, cool, I never knew that those equations were really actually that simple, because they seem so complicated when you see them on the paper. That's my hope. But we can get into maybe more of some of the details.
Sounds great. So I guess the basic idea of an attractor state is really that it's a bit like a valley, and there's a river that then is attracted by that valley by flowing into that, right? I guess that's somehow like, or that's the landscape, I guess.
Yeah. Well, there are many different types of attractors. There are sort of a whole field into themselves, but the basic idea is that you can conceptualize the activity of a complex system as it's evolving over time. And so that means that it falls into what we call a state of a dynamical system. Now, there's lots of dynamical systems everywhere, and they don't necessarily have to be interesting. So walking down the road to the beach near our house, the sand on our beach is a dynamical system. It changes over time with the tide and the winds, but we don't think that it's carrying any information forward or any memory or anything like that. So to say that the brain is a dynamical system isn't that profound. It's a particular type of dynamical system. But if you want to describe any kind of dynamical system, a really good place to start is to ask what kind of attractors are enslaving that dynamical system's activity. And what that means is that, is there a set of rules or a set of constraints that are allowing the system to act in a particular way? And we can imagine that as a valley, and then a ball would be rolling towards the valley. So a classical example comes from genetics. When we're born, a particular gene will be expressed. That's like a valley forming in a landscape. And then where we are before that gene has had its effect is up the top of a hill. And now we're going to roll down the hill into that valley and express the phenotype of that gene. So that's the kind of concept. Now, that type of attractor, this sort of idea of a fixed point attractor, is really only but one of many. There's lots of different types. Another one that people that listen to this podcast might be familiar with by a different name is a limit cycle. Now, a limit cycle attractor is one in which there's a movement which then comes back to the origin. And basically, if you play that movement out, that circle, in two dimensions, it looks like a spike or an oscillation. And so anytime we see an oscillator or a phase synchrony, we often think of a limit cycle as the attractor that's enslaving the dynamics of that system, that's causing the emergent activity to look like, let's say, spiking or oscillatory behavior. And there's many other types. The other one that people may have heard of as a sort of chaotic attractor, where you can have movement in one particular direction, but then with a subtle change, it can then slip into a new movement. The classical Lorentz attractor is a really beautiful example of this. And often chaotic systems have this kind of signature where you can't always tell from where it'll end up. It's not like a limit cycle where it'll always go in the same direction. It can always move into these different surprising directions. And obviously people that study brains really get excited about those because they sort of bake in this idea of randomness or sort of break the idea of determinacy.
So you published an article in Progress Neurobiology this year, and I personally think it was really an instantaneous classic, and in my view, a radical new lead for the field. There was also a lot new to me personally. So after writing such a thought and skillful piece, do you want to retire now? At least that's what I would probably do.
I'm not sure if there's video associated with this podcast, but I'm blushing quite severely right now. It's very sweet of you to say. That was a really, really fun paper to write, and it took a really, really long time. How
did you come up with it? How was the start?
there's a long and winding path to explain the true genesis. I think the proximal cause was that I live about an hour and a half away from campus with my wife and two kids. My wife's a general practitioner, and so I had to come up with ways to pass the time that were productive that didn't involve going into a clinic or going in and working with students. And so I tried to set myself some goals of trying to understand neural circuitry at a level that that I think was available to us from this beautiful work in the field. You know, all these really beautiful new data sets like the Allen Brain Atlas and all this optogenetic studies that are now really refining out the interactions between the parts of the nervous system. So that was the reason that I had the time to really dive in and read. The genesis of the idea really came from a long while ago, back when my oldest son, Tyler, was only just over a year old. And my father, who's an evolutionary biologist, was asking me, about my son. And when he was walking, he noticed that 1 month before he was walking, he was really unsteady and really focused on his feet, like staring down at his feet. And then about a month or two later, all of a sudden he was able to look around the world and he could coordinate and everything was happening really effortlessly. And he asked me what's a really simple question, but in many ways it's sort of really stuck in my mind ever since. And I'm still really trying to grapple with the answer to this question, which is what had changed in my son's brain in that small interval that made him so much more automatic, that made him so much more effortless. And when I went to read the literature, at the time, I had just finished my PhD. I did a PhD in Parkinson's disease, in non-motor symptoms of Parkinson's, predominantly on freezing of gait, but also on visual hallucinations. And so I knew a lot about the basal ganglia, I knew a lot about the dopaminergic system. And I'd always been really uncomfortable with this idea of the basal ganglia as the seat of habits. I know that they're very important for forming habits and for executing particular habits in a particular part of the life cycle. But the idea that you would place the pressure, the computational pressure of an automatic function on something as capacity limited as the basal ganglia never really meshed with me. Part of that was that I came from my clinician supervisor, Simon Lewis, was really interested in this idea of overloading the basal ganglia. We thought of it as a funnel rather than a set of parallel loops, like the classic Alexander DeLong and Strick. We thought of it as this funnel that could be overwhelmed. Its capacity could be kind of short-circuited at particular points in time. And Simon's hypothesis was that freezing occurred because there was a sort of short-term overloading of the circuit. Some crosstalk in the cerebral cortex meant that the striatum could no longer fulfill its role of coordinating movement or helping you to dual task, let's say think while you're walking. And all of a sudden, you got this overwhelming increase in inhibition from the globus pallidus, which would then inhibit, let's say, the MLR or back into the thalamus, and you get this paroxysmal cessation of gait. And so for us, the traditional model of the basal ganglia, sort of this place where you stored a habit, was never really baked in from early on. And so when I went looking around for answers of systems that could support this much more distributed, automatic, precise, high-dimensional responses that you need, you don't have to go much further in the subcortex to find the cerebellum, which is just the perfect architecture for this, right? It's receiving a efference copy of every little output from the cerebral cortex from layer 5 pyramidal cells. It's got an extremely exquisite architecture in terms of the connections. It's very modular, very precise connections into the Purkinje cells that hit both the parallel fibers as well around the cell body from the olive. It has a very particular learning rule, which admittedly is still being sorted out. And then it can actually contact the cerebral cortex back via the thalamus. And then control that kind of feed-forward processing mode. So for me, the cerebellum was it was kind of this eureka moment. Like, oh my God, the cerebellum is this beautiful structure. It's sitting right there. And really, ever since that time, we wrote a paper about this. It's in a Frontiers journal, which was really conceptual about how that might be beneficial for cognition. We can maybe talk about that later. But that idea has been really in my mind for a long time. And I finally kind of had the time to sit down and really read the literature required to etch out the details. And it's actually kind of amazing how these things happen. I was sort of reflecting on this a little bit before going on the podcast. I can actually pin down the moment when I think the solution really presented itself. I was having a conversation with a really talented postdoc in my lab, Eli Muller. We were just discussing, he's a physicist, we were discussing interesting things to model, and we both think the idea of automaticity is just really fascinating, and parallel processing, and we were just really having some interesting discussions. And then I was talking to him, and we were chatting about neuroanatomy. I was sort of catching him up on some details. And I said, the basal ganglia and the cerebellum, they both contact the thalamus, but no one really knows. There's some people think they contact the same cells, and some people think they're different, and, a sort of big hand wave. And then I thought, that we could probably do better than that. And I went and did a Google search, and I stumbled upon this absolutely beautiful paper by Kuramoto and colleagues in Japan that basically traced the cell populations in the thalamus, which we call the core and the matrix populations. I can speak about them in a second. But what they basically showed was that the core and matrix populations, which are identifiable by a calcium binding protein that is expressed individually on each of those different cells, actually differentially receive input from the cerebellum to the parvalbumin expressing core cells, and basal ganglia input from the globus pallidus onto the calbindin cells. Now, the reason this was so important is that I had read before this beautiful work by Ted Jones. Ted Jones is kind of like Murray Sherman level important for the thalamus, right? He's like extremely, like there's two grandfathers of the thalamus, and they both have such important stuff to say. Murray Sherman's approach was to take the lateral geniculate nucleus, primary receptive nuclei from the retina, which then projects on. It has multiple types of cells, but it's predominantly core type cells that project on into the granular layers of V1. It also has some interspersed cells within it, but they really focused on that architecture and really etched it out in really pioneering work. And they would call that a first order nuclei. And then in the thalamus, there's also other nuclei, which they would call higher order. And they characterize those because instead of receiving sensory input, they receive cortical input and then send input back to the cortex. So they're a little bit like the hidden layers in a deep neural net. Right? They never see any of the input. They never get anything to do about the output. What they're doing is they're controlling the context, and they're changing what information will be passed on where. They're kind of like a way station. So that was Murray Sherman's way of characterizing things, very influential. Ted Jones started from the thalamus, and he said, what if we go look under the microscope? What kind of connections can we see? And he described what he saw as a spectrum of types of connections. One of them is the classic core type. They're the ones that Murray Sherman spent a lot of time thinking about. The retina comes in, contacts a core cell into the granular layers of the cortex. Interdigitated and sort of mixed in every nuclei of the thalamus with those are these matrix thalamic cells, and they have a completely different type of connection. What they do is they project up diffusely and hit the supragranular regions of the cortex. I don't know if you have video, but it's this sort of thick area up the top of these layer 5 pyramidal neurons, right?
So we don't have video, but yeah. So the matrix cells contact the apical layer, that dendrites. So the same neuron, but that goes through more or less the whole cortex. But we have the apical layer.
It goes right up the top, and it branches out, and it hits multiple different cells, and it hits them in a region where the cortical feedback from a higher level of the cortex is making its impact. And so this automatically suggests that these two things have very different functions, right? If you have a very different type of connectivity, then you're going to have a different type of function. And now all of a sudden, we can track it even further back. We can say now the cerebellum is contacting one type of connectivity, and the basal ganglia is gating the other type. So all of a sudden, instead of having a bunch of literature that I'd read about the cerebellum, and a bunch of literature about the basal ganglia, and a bunch of the thalamus, and a bunch of the cortex, now all of a sudden I could see a way to knit them together. I could see a way that I could trace the logic of the connections around. And it's admittedly, a very simplified story for how they might work together. But the logic of that interaction was so enjoyable for me, and it was so much fun to think of the implications that I really dove into that and tried to work out, okay, if this is the architecture, why would it be like this? What would it by the system? How would we describe the way that the system's dynamics would evolve if, let's say, the basal ganglia or the cerebellum or the cortex was the predominant controller of the action at any point in time? And this sort of drives us back around to where you get to attractor landscapes, because that language is just perfect for thinking about how the distributed activity of this big complex system can evolve over time in particular ways. And so that's really the genesis of the story. I'm not sure if that's more involved than you were hoping for, but
did the lockdown help to work out the details at all? Did that have an impact?
The lockdown came after the paper got one of the absolute worst rejections of my career. I sent it to a really nice journal, and I was extremely surprised when they sent it out for review. And I received, this was probably going on a year and a half ago, and I got 4 reviews back that basically called into question my credentials as a scientist. They thought that it was too speculative. They thought that it was combining areas that didn't need to be combined. They didn't understand why you would go using the language of dynamical systems when you were talking about neuroanatomy, and why was someone who does functional MRI research talking about cells, and the whole thing. It was just honestly like it was like the old school pie in the face kind of slapstick movies. No, look, academia is one of the best jobs in the world, but one of the prices you pay is that you get humbled intellectually a lot. And I'm either like, I've either got really bad Stockholm syndrome, or I've convinced myself that it's actually really helpful because it causes you to reflect upon your assumptions and to make arguments stronger so that they hold up to scrutiny. And so basically COVID was great because what it meant was that instead of just like saying, oh, you know what, I'm not going to deal with that anymore, I was forced to like really confront what I'd done. And in the end, I think the version that ended up in Progress in Neurobiology is much more mature, much better defended, more refined. I'd been reading a lot of Valentino Breitenberg, who's a real academic influence of mine, and he has this beautiful way of breaking things down to the kind of the nitty gritty of the neurobiology that I just love. And I tried really hard to take what I had before, which was very kind of like, I was painting a picture rather than really etching it out. And I tried to etch it out much more. And it was a hard road to go down. It was definitely like very humbling. Yeah. But I learned a ton through that process, and I'm much happier with the outcome. So yeah, COVID helped in a way.
Great. Okay. Okay. So let's dive into it a bit more in detail now. Maybe just to recap, and I plan to roughly follow the stream of the figures. So talking maybe about, so if listeners want to pull up the article in parallel, speaking a bit about figure 1 now. That is basically reproduced from Ted Jones, whom you already mentioned from his TINS article in 2001. And again, just to recap what you already said, we divide by that the thalamus into core and matrix cells, and these are some cells have both properties, and so it's more a gradient, I guess, between core and matrix. Core means they are parvalbumin positive, and matrix means they are calbindin positive. And they seem to be clustered in the thalamus. So specific nuclei seem to have different, are more core or more matrix. Then core projects to the granular layer, and matrix to the supragranular layer, so layers 1 and 2. And from that, we can also learn, and that's, I think, what's radically new, or what's the centerpiece of your article is the thalamus plays this central role. And the core cells basically receive input, glutamatergic input from cerebellum, sensori nuclei, and the cortex. But cerebellum is key here, I think. Then the matrix cells receive input from the basal ganglia.
Yes, that's right. So, you'll probably already tell listeners that this is a sort of very detailed neuroanatomical argument that I'm trying to make. Yeah. But my hope is that by laying out things in detail, people can go and read the source material. Definitely. Go read Ted Jones. Go read Helen Barber's work on the cerebral cortex, the structural model, and read about these really beautiful constraints and patterns that have been kind of etched out by these really brilliant neuroanatomists. And another one that I'd recommend is Francisco Klaska, whose work is just beautiful. He's sort of like, and I've told him this to his face, he's sort of like the reincarnation of Ted Jones to me. He's he's really etching out this beautiful, dizzying complexity of thalamocortical connectivity that really kind of captures this idea that Andy just mentioned, which is that every nuclei in the thalamus, we traditionally, when we look at it, if you look at a Google search of the thalamus, you'll see that people will often chop it up into subnuclei, like the anterior nucleus or the mediodorsal nucleus, the lateral geniculate nucleus, the pulvinar. But if you go down and look in the actual sort of cellular compartments of these cells, there's actually much more of a blend of cell types. As Andy said, some of them are predominantly of one type, but almost all of them, I believe, share a blend of both. The one exception to that rule are what we call the intralaminar nuclei, and they are almost completely matrix type, and they're kind of like a subgroup of the matrix type. So I haven't mentioned it yet, but all of the matrix nuclei project also to the striatum as well. And the intralaminar nuclei are extremely likely to project to the striatum, in fact, much more so than to the supragranular regions of the cortex. But yeah, there's this idea that the thalamus is actually, instead of being a set of little kind of like modular nuclei, is instead a kind of smear of different cell types, which are then projecting onto the cortex. And then the way in which they project onto the cortex, based on the logic of the cortical connections, can then tell you about the kinds of different processing that could occur when the different thalamic nuclei were recruited.
So maybe I think for the listeners of this show, the human motor thalamus is the one they know best. They would know best. So I guess in this logic, the VA nucleus, for example, would receive input from the basal ganglia. So it's likely a matrix type nucleus, right? And the VL or VIM would receive more the input from the cerebellum. So that would rather be a core thalamus, right? Can we say that?
Yeah, and I think that's fair. There are exceptions to that rule as well. So biology is, if anything, is fickle. Laszlo Aksadi's done some really nice work showing that the output of the cerebellum, the deep cerebellar nuclei, do contact the intralaminar nuclei. They also send connections to lots of kind of weird places like the locus ceruleus and the dorsal raphe and the ventral tegmental area and all kinds of other areas as well. So there, in a way, you could think of the kind of logic of the organization is kind of predominantly in one way, but there's lots of exceptions to the rule. So I don't want people to come away thinking that this is like the way it is organized.
Absolutely.
But rather, this is one of the sort of major backbones of connectivity that's present.
We still need to simplify a bit, but I totally agree with you. And if you look it up, everything seems to be connected to everything in the brain always. So there is more detail to each level. I totally agree with you with that.
So one of the things that's really fascinating to me, and probably just to kind of steer this in a direction that your listeners might be interested in, is that the fact that the basal ganglia cells, so the striatum inhibiting globus pallidus internus and globus pallidus internus, and then sending GABAergic projections down to the thalamus. The fact that those cells are matrix type predominantly and not core type was really contrary to what I think the PhD version of me would have expected. I think if I was taking the Alexander DeLong and Strick model, which I think has been extremely influential and has, again, I don't want to feel like I'm arguing against that model. I think it's got a lot of logic to it in the idea of cortical regions in a distributed fashion, sending projections down to specific locations within the basal ganglia, where orbitofrontal is to the accumbens and dorsolateral prefrontal is to the caudate and motor to putamen. The logic is all very consistent. The segregated pathways within, again, consistent. There is a collapsing of the dimensionality, which I think is a really important feature, which we can talk about if you'd like. Charlie Wilson at UTSA has done some really beautiful work on that. He has a paper in the neuroscientist called Active Decorrelation of the Basal Ganglia, which is one of my favorite papers on the basal ganglia, which we can talk about if you like, which talks about that collapsing nature. But then the really important feature is that the DeLong and Strick model, the Alexander DeLong and Strick model, was always, the analogy that was brought up is this idea of gating an action plan. The idea would be something like if your dorsolateral prefrontal cortex innervated the caudate could then silence a particular globus pallidus neuron, which would then lose its inhibition over a thalamic region, which would then allow a synchrony between the cortex and thalamus. And it could, let's say, oscillate along at some high frequency which would mean that it had an impact and some other area didn't. That idea of thalamocortical synchrony is sort of inconsistent with this idea of the way that the matrix thalamic nuclei project back to the cortex. They're not coming back and hitting the same neuron that contacted the striatum and then sort of opening up the gate. They're projecting back in this, I'm going to call it diffuse. I'm doing inverted commas with my fingers here. It's a diffuse asterisk.
Yeah.
So it's not diffuse in the sense that every single neuron in the brain gets contacted.
Sure.
But it does come up and it projects to the supragranular areas in a relatively diffuse fashion.
Yeah.
Such that the region of the cortex that innervated the caudate, let's say, to open up that channel, will get a little bit of a boost. Its feedback signals will be now primed. You could say the gain has increased in those signals and made them more excitable. But so will a range of other regions around it, another cortical areas. And that means that you're sort of almost, in a way, losing control over what you've done. In a sense, the basal ganglia is opening up the channel such that now more of the cortex can have a vote in what the ultimate action is. And I think that has some really profound implications for how we think about what the basal ganglia is doing for normal behavior, but also what happens when the basal ganglia becomes dysfunctional, let's say in Parkinson's disease or Huntington's, or a number of other sort of
Before we get into that, and I think that's really the central point, right, that the basal ganglia seem to broaden the attractor valleys. But before we go into that, I think let me try to dumb that down even more. I know.
Please do.
We're simplifying a lot. I know that, and everybody should be advised to, of course, read the original material. But just for understanding, so I guess what you're saying is the classical Alexander DeLong Strick model would usually be conceptualized in talks maybe for med students that there are different types of actions in the brain, in the cortex. And then, one is selected, the action selection thing. And that's being done by disinhibition via the GPI, this action is now passed through and done. That already is a very, very strong simplification of their idea. But that's, I think, how people see it. And your model would propose instead would rather be that the basal ganglia add some sort of randomness into the model, and that the specific region of the cortex that was starting the whole thing gets more feedback from other cortical areas that are close to it. Does that sound right? And let's maybe try to shape out once more why that is. So it's because of the matrix cells in the thalamus diffusely project to the apical dendrites, while the core, and we haven't talked about the core that much, which comes rather from the cerebellum, would rather somehow via some interactions with stellate neurons and go to more a specific region, right? A feed-forward type.
Yeah. So I think everything you said was right on track. So I think to help understand why it would matter that the cerebral cortex was perturbed either in a precise granular layer or in a diffuse supragranular layer, it helps to sort of zoom out a little bit and think about the organization of the cerebral cortex. Now, the cortex is a thin sheet, approximately 6 playing cards thick. It's all convoluted and smushed inside the skull. And, we're still understanding the mysteries of the cortex, but a simple way to think about it is that there's one end that's tethered into the external world. The retinal inputs hit V1, the auditory inputs via the cochlea hit the auditory cortex, the sensory inputs via your body hit S1. They're the tethered areas. And there's all the way at the end of the other end of the connections are the what we call the cingulate cortex, or areas in the orbitofrontal cortex, or M1, the movements. So along those two extremes, the cortex changes drastically in the way it's organized. On the sensory end, it has this really thick granular layer, which is where all the core cells are coming in. And it's typically thought of that they're a feed-forward layer. What that means is that an input will come in and they'll project to cells that are what we call the supragranular layer, then propagate forward up towards the more motor or cingulate regions. At the other end, there's much more of what we call output cells. They're in the infragranular layers, layer 5 in particular. And if we're in the motor cortex, a layer 5 cell would be the BET cell that project down to the spinal cord and made contact with alpha and gamma motor neurons. It can move muscles and make very precise refinements to movement. And the idea is that this is an idea that's been around for a really long time. Classical work was done by Fellman and Van Essen, Pat Goldman-Rakic. Some of the more modern work, Helen Barbas and Bas Sikopoulos, have shown this really beautiful patterns where the sensory regions, which we call granular cortex, the rule is that if the inputs are received by that area, they'll send a feed-forward projection to a granular layer of a higher level, a region that's closer towards the motor end. In contrast, a motor region will send a projection that feeds back to the lower areas, but instead of contacting the granular layers, it actually ignores them completely, and it sends projections to the infragranular layers around the cell bodies, and also to the supragranular layers of the cortex, this really big, thickened area that's full of dendrites. Now, what are dendrites? Dendrites are the connections that are sitting on the ends of cell bodies that receive the inputs to a neuron. And one of the really fascinating things that happens in the mammalian cerebral cortex, and it's really kind of expanded and it's really augmented in humans and in non-human primates, is that the big output cells of the cerebral cortex, these layer 5 pyramidal cells, have a really elongated stalk. So the cell body is sitting there in layer 5, receiving feed-forward inputs from the lower layer, but it's got a really long tube, a long stalk that connects it to its feedback from the higher area of the cortex. And what that means is that because the stalk is so long, there's just so many ways for the message to get lost. You can imagine that if you had a long walk to the post office, and people were sending you letters, if your walk was treacherous, there's just every likelihood that you turn around and go home a bunch of times before you finally got to the post office to receive that message. And so what really happens then in the cerebral cortex is absolutely fascinating, is that if those messages are received in short order, or if there's neurochemicals around like noradrenaline or acetylcholine, the cells can get those messages can get pumped down to the cell body, and they can cause the cell to function in a completely different mode. The idea is that when there's only feed-forward connections hitting the pyramidal cell, it just sends a spike. But when the feedback connections come to, what they do is they basically cause a huge increase in calcium that then causes that cell to burst. And that bursting can have a really big change, a big effect on ongoing dynamics. It can change the way that the cell's forming, the attractor landscape is forming. Now, why is this important? Because the basal ganglia now has control over whether or not a particular cell receives its mail, whether a particular cell shifts from a spiking into a bursting mode. And it doesn't do it in a way that just causes the cell that sent the message down to the basal ganglia through the thalamus to get its mail, but its entire neighborhood. So what you've now got is a different way in which the cerebral cortex is using the input of the basal ganglia to inform its ongoing brain state, which of the different cells are firing in that burst firing mode, in a way that is not completely inconsistent with the Alexander DeLong and Strick model. It still is going to gate the function of that particular area, but it does it in a way that also buys in some randomness. The fact that your neighbor could also get their mail when you went to check for yours means that the neighbor might get some mail that might change their behavior, and the whole system can now evolve and do what we would call almost like a swerve from what it would have had before. And I think that's really fundamentally important when we think about our behavior and the importance of chemicals like dopamine and the architecture of the basal ganglia for informing that kind of behavior as we sort of change over time.
Super. And I think that's a really crucial concept that you just mentioned. I think that was established, among others, by the group of Matthew Larcum.
Yeah.
You have the same neuron, PT-type neuron in the cortex. If you just give it input to the basal dendrites, it fires a single spike. If in a specific time window you also activate the apical dendrites, then it goes into a bursting mode.
Yep.
Is that correct?
Yeah, that's right.
And that bursting mode is, according to your model, more or less activated by input from the basal ganglia, right? So the basal ganglia would recruit that one single neuron, but also its neighbors to somehow go into a bursting orchestra.
Yep.
Is that correct?
Yeah, that's the idea. And I think this is where it becomes kind of interesting. That kind of a mechanism is a really great way to almost hedge your bets. Let's say that you decided that you want to learn how to shoot a basketball, and you take a shot and you miss it, and then you take a shot and you make it. Well, part of the architecture of the movements of your muscles that caused you to make the shot was beneficial, and part of it was completely terrible, and part of it was you missed out on the ideal, let's say, angle of your elbow and the ideal timing of the contraction of your triceps and relaxation of your biceps, let's say. But in a way, because there was an outcome that was positive over time, the idea is that the system can have some of that variability required for optimal learning. You don't have to be spot on perfect every single time. What you have to do is get experience and feedback and continually try to improve. And by having that variability baked in, if we go back to the attractor landscape idea, if there's a perfect idealized attractor somewhere that represents the perfect basketball shot, let's say Steph Curry's jump shot, and your ability to kind of like coordinate all your muscles in the correct way to shoot the ball, the being variable gives you the amateur a much better chance of finding that perfect attractor than it would be if you just tried to optimally do it every single time, and where you're sort of poised on a knife edge. And so it's very hard to test out these ideas. But one area of empirical research that I think is really informative is birdsong, where people understand the importance of the basal ganglia for these beautiful songbirds that are born and hear their father's song. They try to mimic the father's song. Early parts of their life are really it's extremely important that they learn how to sing the family song. But then later in life, as they become juveniles, it's actually much more important that they deviate from that song, kind of like Miles Davis playing a solo. They need to be able to come up with their own progression of the song that's unique enough that they can stand out from their father. And it turns out that the basal ganglia is actually incredibly important for both of those things, right? It's important early on for learning how to mimic the song. It's also important later for deviating upon that and actually soloing and improvising. And I think that this architecture, this contact between the basal ganglia and the matrix cells offers a mechanism that could plausibly buy you both. It's slightly less efficient, let's say, as a learner, but it also has a longer term efficiency where it'll find the optimal solution more often than if you try to very precise outcome and only execute the precise outcome with high validity every single time. So it's a slightly gnarly concept. It's really a systems level way of thinking about the brain and how it's functioning. But I think it's much more parsimonious for the kinds of ways that we learn, the sort of noisy trial and error and then improvisation that I think is a real characteristic of how we learn skills and habits.
Definitely. So I guess that really poises us now to talk more deeply about how this all fits into attractor landscapes. And I guess we're now at figure 3 around that, right? And you show that basically, again, cerebellum and other systems would activate the core, which gives a single spike, which maybe forms the attractor landscape in one way, but then if you just hit the matrix, that doesn't result in spikes. So that's the apical dendrites, but it rather broadens the attractor landscape a bit. So that's more the basal ganglia input. And then if you do both, you would get the bursting mode, and that would really make a deep valley of that attractor state.
So part of my sort of long winding road that I mentioned before, after my postdoc with Russ Poldrak, where we had done a lot of work on brain networks and human brain imaging and trying to understand some of the rules that govern how brains can kind of change over time, I started working with Michael Breakspear, who's at the University of Newcastle here in Australia, using what we call neural mass models to try to model brain activity and try to think about the kinds of rules that go into governing how the brain can change over time. So remember from earlier, I talked about how the brain is an example of a dynamical system, and it's a really interesting type of dynamical system. It changes over time in particular ways. Well, the equations in the top of figure 3 are some of the kind of ways that we approximate that change over time. It seems complicated. Don't forget, I have a clinical training. I still feel slightly ill every time I see an equation. But the idea here is actually relatively simple. If I want to ask how will my region, let's say X, change over time, the little dash that you see after the X just says, how is it changing over time? What is its rate of change? All that I need to do is I need to find out what it was doing the time point before, and I subtract something like it's how much it will relax. That's the tau version, the minus tau. I then the big E is the sum. What I do is I sum all of the inputs from all of the regions it's connected to. That's the AIJ, that's its adjacency matrix at the previous time point. You could think about if I've got a bunch of friends, they're all egging me on to jump off a high diving board. The second before, if 20 of my friends all say jump, I'll be much more likely to jump the next time. However, the sigma parameter is then you could think about how much I'm listening to my friends. So if I decided that I wasn't listening to them all, sigma 0, it doesn't matter whether they're talking, I just blocked them out. If sigma's 1, even one of them could talk and it would be enough to make me jump. And then the final thing at the end is the E parameter. That's just noise. That's just saying that there's other stuff in the system that we haven't modeled. So it's actually really simple. But one of the things that it lets us do is that it turns out that that equation that we have on the right side of that equation is actually empirically the same as talking about an attractor landscape with a ball rolling around the landscape. So we could think about X being the ball, and then the way that the X moves is the tau parameter. The inputs it's receiving is how much of a kick the ball receives to move in a particular direction. And there's also a little bit of noise. So this is where I sort of mentioned before that if you were coming at the field to the field of complex systems, as a newbie, it's incredibly overwhelming. There's all these new terms and equations, and everything seems, really stressful. But if you realize that you can get a lot of your intuition from this idea of a ball rolling around a landscape, I mean, we've all seen, a ball rolling down a hill, and we all, really intuit the idea of gravity. So these things can kind of help, like bootstrap you up. So the idea was, if we were thinking about the ball rolling around a landscape, and then we thought about what would happen if the basal ganglia got involved in the ball's movement, or the cerebellum got involved. But I think it's relatively easy to imagine what would happen. If the basal ganglia got involved and you got more apical input, that'll widen out the basin. And if the apical input then caused those cells to change from a spike to a bursting mode, that's going to deepen the attractor down, right? The burst is going to mean that that group of cells has much more influence over the brain. That would be like turning the sigma parameter up on that particular subset of the neurons that received that boost. And so that means that the ball will be much more likely to roll into that attractor before it then dissolves. In contrast, one of the things the cerebellum is really good at is actually conditioning on its own output. So if you feed the cerebellum a context signal like shoot the basketball, and you do it enough times, the cerebellum will learn that every time that you actually bent your knees, then half a second later you popped your knees up. Then a third of a second later, that's when you needed to lift your arm, and then another quarter of a second is when you needed to flick your wrist. It can start to learn that sequence. The way it does that is quite fascinating. The output of the deep cerebellar nuclei of the cerebellum actually send a projection back to the cerebellar cortex. And some beautiful work by Michael Mork's group in Texas have actually just shown that you can use that circuitry to learn arbitrary sequences of inputs. And so putting those together, the idea is that the cerebellum will slightly deepen the attractors, but it'll do it in a really brief way where the attractors are almost kind of coaxed along in time. So with the basal ganglia, you'll get this big deepening of the attractor that'll maybe last for a little while. Whereas the cerebellum, you'll get a deepening, but then it'll move on, almost allowing you to sort of flow through the landscape much more quickly than you would have with the basal ganglia. And so this was really just trying to give people an intuition about what I see as the importance of this architecture, this logic. Because at the end of the day, I'm really inspired by the work of James Houck, who unfortunately passed away this year, which is that we can't think of any part of the brain as existing in a dish. The cerebral cortex is beautiful and complicated, but its computations are only as good as the information it's fed, and only as good as the feedback it's receiving from the thalamus and the basal ganglia and the cerebellum and the colliculus and the hypothalamus. It's only in thinking of the system as a whole that we gain the understanding of the algorithms that it's trying to solve and trying to embody. And so for me, this was my way of trying to say, I think there's a really clear logic of cortex, thalamic, basal ganglia, cerebellar connectivity. And this is how I could see it playing out at the level of the system, in the idea of it coaxing the ball around the landscape in a slightly different way.
I mean, to comment on that, your paper is really one of the rare examples that goes from cell to systems level neuroscience. And that's what makes it so precious, I think.
Well, thank you very much. I think I have probably a lot of gray hairs to show for all of the furrowing of my brow that I've done trying to think about this stuff. But I think it's really rewarding. And to sort of put a shout out there for systems neuroscience, I think this is a really exciting time for young scientists. So if you're coming out of undergrad and looking for what to do, it doesn't really matter whether or not you trained in coding or in physics or in neurobiology or in psychology. If you're curious about how the system works as a whole, there is so much data out there now, so much information, and it's like way too much for any one person or one group to do. So it's a really great time if you like complex problems and trying to think about high-dimensional variables and how they interact with one another. Kind of akin to what it was like before the Human Genome Project came along, right? Where people were starting to get a grip on start codons and stop codons and our mRNA and tRNA and rRNA and all of the players, and then starting to work out how they actually work together to fulfill the function that's sort of bigger than all of them. That's the time we're at neuroscience and systems neuroscience right now. I think it's a really exciting time for young scientists in the field.
I couldn't agree more. We are currently in a situation where we have so much data, more data than we can probably analyze. And I think I've heard it at least 2 times in the Brain Inspired podcast that what we currently need is theory, right? Not so much more data, probably. So I totally agree. There's so much to do in this regard. Let's talk a bit more about the cerebellum core system. I think you mentioned to some degree, and I'll cite you there, that the cerebellum can be conceptualized as acting like a storage center for the spatiotemporal patterns that have been learned over time to instantiate well-learned behavior. And it's really, the idea is that you have a specific particular context, and within that particular context, such as throwing a ball, but each part of that movement, even that particular context, it is really good at predicting what will likely occur next, and then doing what is needed to next, right? Is that correct?
Yeah. So I'm really inspired by a lot of really brilliant thinkers about the cerebellum that have come along before me. I mentioned Hauck and Wise before. The Lena family, husband and wife combo, did some beautiful work. John Montgomery, Kiwi, just across the pond from me in Australia, has done some really beautiful work thinking about the implications for the cerebellum. And John's work, I think, I wasn't aware of it when I was doing the analyses and thinking back in the original Frontiers paper with my father. But I came to John's work later, and I think he has a really beautiful way of putting what the cerebellum does. And it comes from the cerebellum's evolutionary history. And it turns out that the first kind of known sort of time that the cerebellum popped up was right around the Cambrian explosion, when all of a sudden in the water there were these mega huge sharks and teleos fish that could basically hunt and take out their prey. Big animals, and to move in the water for them was a huge cost. And so they needed neural structures that were really, really good at anticipation. And they also needed good structures, cerebral structures that could cancel out the inherent noise in their big, lumpy bodies and systems and give them what was left over, kind of like noise canceling headphones. And through a remarkable feat of evolutionary engineering, the cerebellum is really wired up to do precisely that. It's wired up to take a particular context signal, subtract out what is known, and leave the remainder, this sort of active noise cancelling. And in the context of the human system, which evolved over time, and the kinds of inputs we feed it, it's really good at anticipating what people call the sensory consequences of motor action. So sometimes you'll have heard of this as called an efference copy. The idea is that every time you move your arm, let's say, you reach out to grab a glass of water. As you reach out, there's a bunch of signals that your brain is sending out saying, moving to location X. And, that requires moving this muscle and that muscle, this particular tone. What the cerebellum is really good at is saying, the last 10,000 times you did that, here's the thing that worked really well to ensure a stable transition.
Yeah.
So it stabilizes the movement by anticipating ahead what ought to be coming so that they can prepare the nervous system. And I think this is actually extremely crucial. If you were a shark trying to hunt your prey, if you could anticipate its movement, you could get there before the prey got there and you could eat it. If you are a human and you're trying to shoot a basketball, if you can anticipate how the basketball will feel as you lean forward to shoot the ball, you're going to be that much better at shooting the basketball than your compatriot. And I would go even further than that and say, if you were playing chess and you can anticipate the way that your opponent would react to your movement or your perception of their movements, corollary, what have you, to some large level of abstraction, you're going to be much, much better at beating your opponent because you've been able to automatize the anticipation. And I think that's incredibly important. It's a really beautiful structure of the cerebellum. So this idea of adaptive noise canceling and prediction of the next step, anticipation, I think are really crucial for what it does.
And that is really central because the brain is a prediction system, right? Mm-hmm. I realized that quite recently only that if we train an AI currently, a neural network that has to, classify, for example, apples versus oranges, then we usually use labeled sets, right? We have, we give it a set of 1,000 brain images where we say this is an apple, this is an orange, and then it can just make sense out of that. But when we grow up as toddlers, as babies, we don't have such sets of labeled data to learn from. So I think one of the few Yes, noisy parents, exactly. And one of the few strategies without labeled sets is really to introduce time, to have temporal difference learning, where we, do something, we get a result, and then we over time form or are able to form predictions by repetitive doing that what will happen next, right? So we all of us have seen toddlers throw plastic cups to the ground, and we ask ourselves maybe in the beginning, why do they do that, right? But they just want to see what happens, right? So they explore their environment, then form predictions. And then there might be, of course, what happens can be in prediction error when, the model needs to be updated because you see something that is not expected. One example could be that it is not a plastic cup, but a real cup, and then it shatters to the ground, and the toddler needs to update its model. So I guess in that regard, and we are slowly migrating to figure 4 now, your novel insight with the cerebellum has also, and I think that involves the basal ganglia as well, right? So the orange part of the figure is basically the basal ganglia's input. So can you contrast the older view, which is more the cortical route, with the cerebellar route of that figure?
Yeah, sure. So this last section of the paper was really my attempt to zoom out a little and say, okay, if you've joined me on this journey, you've learned a bunch of neuroanatomical terms that you didn't think you'd ever care about, and then I force you to learn about dynamical systems and all of the language that goes with that. Why would I do that work? And my hope was that in doing that, by seeing the system with new eyes, you could see how could it achieve some of the things that we think are really crucial functional capacities of our brains. As you've mentioned, one of them is predicting ahead, anticipation. I think as we stand here in 2020, there's very little pushback on the idea that the brain is somehow instantiating some form of what we would call predictive coding or predictive control over the environment. The idea is that if you expect to see something, you're sort of changing the way that you interpret the incoming input. There's always a hypothesis and then a collection of evidence and then an ongoing refinement between those. And that's a better model for perception than, say, just a camera taking a photo and then registering where the pixels were dark or light. So there's this really different way in which the cortex is is functioning. And there's been a huge amount of really beautiful, elegant work in that space, looking at how is it that the cerebral cortex could instantiate this idea of hypothesis or a prior, which is the in the language of Bayesian mathematics, and then the posterior being the extent to which the prior, your guess, matches the evidence that came in. If they match really well, then the posterior is really, really, really big. You're like, oh, great, I expected to see my friend's face and I saw it. But if you have an expectation your friend will come and then someone else walks through the door, then there's a big error, and they call that a prediction error. So one of the things that I always found a little frustrating is that a lot of those models were really based on an understanding of the cortex as the brain. And I think not to throw shade at the field, but in a lot of ways, a lot of what neuroscientists have done over the last little while is really sort of take the cortex as the seat of all the interesting behaviors in a brain. It's really obvious in mammals. It's expanded in humans. It's right there on the outside. If you have a lesion to it, let's say a stroke or a tumor, you get really specific localized impairments. So there was a lot of logic going towards this sort of conclusion. But then there's also evidence to the contrary, that you can have, decorticate animals, in experimental settings. Animals that have their cortex removed will often be much more active and move more. This is the work of Jaak Panksepp. There's also really fascinating syndromes like the Sprague effect, where cats that have a lesion to V1 are functionally blind until they perform a small lesion right near their superior colliculus, and now they become sighted again. So there's all these really fascinating descriptions of the sort of primacy of subcortical, or at least let's just say the importance of subcortical structures for a lot of the functions that we think mammals and humans are capable of. And so I've really had that in the back of my head for a really long time. And one of the things that I always got a little bit annoyed by was that the cortex, might send a prior or an expectation, but what if some of the ways that it's getting that expectation is because of some of the subcortical architecture, like the basal ganglia, the thalamus, the the cerebellum, or the colliculus. And I was just trying to play out the idea of, well, if anticipation is what we care about, here's this great structure, the cerebellum, that's set up to do anticipatory functions and do it in a way that the cortex is really bad at, which is with high precision, really, really quickly, and do it in sequences with really, really high avidity. In a way, it's a little bit like those classic player pianos that you might have seen in the old spaghetti westerns, where the piano's playing itself. There's a little roll, a little set of bumps like braille, and they play out a particular song, right? It's not going to be able to deviate from that particularly well, but it does it in a way that will play it out with really high precision over time. And so I think, in a way, what I'm trying to do is just make the argument that if we think about the benefits of the subcortex, maybe they're going to help make our priors better in a different way. They're going to make them quicker. They're going to make them slightly more automatic, let's say. They're going to help you see the outcome and then rely on your knowledge of that outcome so you can focus on a higher detail. With the basketball example, I don't have to worry about where my elbow is. I can focus on whether or not a defender's coming in front of my face, or whether there's enough time left on the clock. Whereas if I didn't have that automatic function, I'd have to be worried about whether my elbow was wobbled into the right angle before I shot the ball. And so I think, in a lot of ways, expertise really is making these really automatic predictions or priors, rather than the really deliberate, explicit, specific priors that we make via the sort of what I've described as the cortical route.
Would that still be involved if you learn something completely new, or would that really be once you've learned it, you delegate it to the cerebellum?
Yeah, it's a great question. I mean, part of this is, the stuff that's to be worked out. My guess is that the first time that you see something novel, your cerebellum is trying its best. It's sort of like the helpful friend. It's like, oh, maybe you should try this. And, if it's, if you just gave me like a, let's take an example, curling, right? I've never curled in my life. And someone goes, here's a curling broom, go have some fun curling. I'm going to be out on the ice brooming my best curl. And I'm sure if you asked an expert curler, they'll say, actually, it's a little different than sweeping with a broom. You have to do this, this, and this. But to me, my cerebellum would say, you have a broom-shaped object. They're trying to sweep. You should just do the broom action, right? And so to me, the cerebellum is always going to be giving guesses for what might work. And this is where it kind of gets real fun, much more speculative. But this kind of starts to have the flavor of what we would call intuition, right? Sometimes people have anticipatory behaviors that they can't quite put their finger on. It's not something that they consciously thought, oh, I will do A, B, C to achieve Y. They just feel like they ought to do it. And one prediction might be that the cerebellum and maybe other structures that are really set up to automatize, like the hippocampus, are sending out priors that say, hey, this might be a good behavior for you in this current context. Even though you haven't joined all the dots, you haven't noticed the elephant in the room that, something's happening, you ought to act quickly. And I'm really compelled by that. And I think it's a much harder idea to test empirically. But, as you sort of mentioned before, we need the theories first to be able to make the predictions that we can then go out and test.
You mentioned the Frontiers paper in 2014 that you wrote with your father, Richard Shine, who is, as his brother John Shine, as I've read up, a famous scientist and fellow of the Australian Academy of Science. I think both received the highest top awards of Australia for research. And how was it like to work with a father on that? And maybe how did having such an academic background shape the way you looked at these things?
Yeah, it's a good question. So it's really funny, right? My dad and my uncle never really kind of put pressure on me to do anything with science. They're both really, I think in a way science traps you in your own little world. And, if I ever asked my dad or my uncle about what they were doing, they would tell me, but they weren't offering it up as much. And I actually noticed myself doing that with my children a bit. I think I try to bring a scientist viewpoint to them, which is to ask questions, look at the evidence, try to arbitrate between different options for how to explain the evidence, hold multiple hypotheses in mind. In fact, that's actually one of my favorite things about the scientific viewpoint is that you don't have to be so sure of everything. You're sort of trained in a way to be uncertain and to be comfortable with that uncertainty. Whereas I think a lot of the problems that we kind of get into in the world is that everyone needs to be so certain that politician's right and the other one's wrong, or this is the way to act and this isn't. And I think in a lot of places, there's just not a clear answer for what the right way to act is.
A lot of gray in the world. I agree. Yeah.
There's a lot of gray, and I think we need to embrace that a little. But anyway, that's a side point. Honestly, working with my father on that paper was one of the highlights of my academic career. My dad is an incredibly deep thinker and unfortunately has spent most of that beautiful brain thinking about reptiles like cane toads and snakes, which is a real shame because brains are so much more interesting. And so I finally, with this project, got a chance to kind of see my dad in action. And that as a son was just, like a real treat and something that I truly cherish. My father actually also is a co-author on a paper I did a couple of years ago with a few other of my academic heroes, Olaf Sporns, Russ, my postdoc supervisor, and Mike Breakspear. And my father also still has the ability to make me feel like a 7-year-old, who's done something wrong. I still remember him sitting across the table from me, interrogating me about the statistics that I'd run on a particular analysis, and just feeling like I was about 7 years old, like I just tattled on my brother or something. But no, it was absolutely magical. Great. So much fun.
Going back to your paper for a final round, I think, and I must say the paper wasn't at all complicated.
Oh, thank you very much.
Well, it was. But I guess we're moving to the final figure 5 now. In 2013, Nobel laureate Daniel Kahneman wrote an influential book that most of you will have heard of, entitled Thinking Fast and Slow. Do you have anything to say about that?
Yeah, sure. Other than that, you caught me in a moment of hubris. I apologize for the complexity of the paper, but in a way, I was trying to kind of capture what I saw as a kind of way to integrate across fields that I think is really exciting.
And it needs to be complicated. It's amazing.
Yes,
yeah,
it needs to be
that as critique. Yeah.
To Kahneman's work. So there's a whole field of ideas that people describe loosely as dual process models. So the idea being that a lot of what psychology focused on in the early days, because it's much easier to test, is the sort of serial moment-to-moment chunk of stuff that we're aware of. I'm going to flash you a letter. What letter was it? You tell me the letter. I'm going to give you a choice between the blue and the red box. I'm going to give you $1 for the blue and 75 cents for the red. And, you have to push the button with the blue, right? The thing that you're aware of is the thing that they're trying to measure. And so a lot of psychological theories are really focused on that seriality. But if we stop and think a little bit about the kind of construction of our mental lives, there's so much that happens subconsciously. Right? This goes all the way back to Freud and even further back, even to Plato and Aristotle. So the idea of what was the shape of our cognitive life. And Kahneman, I think, really beautifully summarizes this idea that there's these different kinds of modes in which our system can act. And he calls them System 1 and System 2. Mm-hmm. System 2 is the traditional one that psychologists have studied a lot. The small chunk that you're aware of, the serial, slow one, yeah. Deliberate process. Yeah. Under the hood is a really rapid, fast, precise, really what people would kind of call it automatic parallel processing architecture. We would call System 1. Yeah. System 1 is quick, but what it buys in quickness, it trades off in accuracy. Yeah. It's going to make heuristic errors. It's going to be the one that jumps to conclusions. It's going to be the one that sees a shadow and thinks, oh no, someone's coming to get me. Yeah. When really the shadow was caused by, a tree that had fallen down or something. And then the idea is that you get a System 1 input and then the System 2 can come through and kind of clean up the details. Again, as I sort of mentioned before, what I was trying to do in this last section was kind of like re-emerge out of the muck of neurobiology and dynamical systems to say, okay, what can we now say about things like predictive processing, but also things like architectures of cognition, like this dual processing theory. And I think there's a really strong analogy here between the mechanisms we talked about and System 1, System 2. System 1 really is sort of strikingly like what we've described as the cerebellar mediated system. Yeah. Every time you act, you now get an anticipation of what will come next. And that anticipation can be a real quick guess at what might be coming. It can make you really adapted to the environment, or it can make you incorrect. If you have errors, if the system is in a heightened state of arousal, you're much more likely to awaken all those apical dendrites we talked about before. Maybe the basal ganglia does it in a targeted way. Maybe the locus coeruleus and the raphe can do it in a slightly more distributed way. But in doing so, what you do is you then recruit more burst firing, and you make it more likely that more slow, deliberate process will actually occur. We've got some modeling work coming out from the lab, hopefully soon. Brandon Munn, a really talented postdoc in my lab, has been doing some really beautiful work showing that modeling that apical amplification of those layer 5 pyramidal cells can kind of recover many of the different signatures that we associate with consciousness and arousal. Yeah. And we've been chatting a little bit with Matt Larkham, who did a lot of the really beautiful empirical work there recently, and trying to refine that model and make it sort of capture even more of the phenomenology that they see when they look under the microscope. But really, rather than saying that this is the System 1 and System 2 in action, it's really to sort of show you that by appreciating the neurobiology and appreciating the way that it could shape system dynamics, all of a sudden these things that seem like completely different, like predictive coding and System 1, System 2, and Dan Dennett's idea of fame in the brain for consciousness can be kind of looked at through the same lens, and you can start to ask, well, if consciousness is related to a subset of these little pyramidal cells burst firing, well, now the basal ganglia becomes really important for controlling which subset is active when. And now, if you're in a motivated state with high dopamine, or let's say a less motivated state with low dopamine, or if you have Parkinson's, and then you've lost the dopaminergic inputs to the basal ganglia, or maybe you over-medicate, these all now have an impact on that circuitry. And instead of just thinking about it in the realm of movement figure, we can now start to extrapolate out and ask about whether or not people's with Parkinson's have a different conscious experience. And so this is where the loop really closes for me, because it comes back to my world of thinking about hallucinations, sleep dysfunction, cognitive impairment, freezing of gait in Parkinson's, and trying to work out how can we, draw a more complete model of what's happening in the brain for those people so that we can make better treatments. So we can, so I can come to someone like you, Andy, and say, hey, maybe we ought to stimulate here and not there.
Yes.
That's the real goal for me at the end of the day.
So I guess this paper really is a new mechanistic backbone for the things you're usually doing, right? Maybe one could say it like that. And of course, will probably be refined in the future as well. So system 2, the slow one, the thinking, conscious, and so on system in your model involves several regions of the cortex to transition into this burst firing, right? And that is being directed maybe more or less by the basal ganglia. Is that correct?
So Matt Larkham's done some really beautiful work where there's sort of 2 lines. One of them, they take these layer 5 pyramidal cells and they place the animals under different forms of anesthesia. They use a couple different gases. I think there's like one with isoflurane. There's another one with they give propofol. Okay. And they show that the apical dendrites decouple from the cell body. And then that's one line of evidence to suggest that this function of those cells is really important. The second line is that they actually record from awake animals in the, I think it's barrel cortex, but it could also be visual cortex. Don't quote me on that. They have a really beautiful Nature paper on this. And they put the animal into a condition where the visual or the sensory input to their whiskers is at threshold. And what I mean by that is on 50% of the trials, the animal will report that they felt or saw the stimulus, and on 50% they won't. And so the idea is that it's like a light touch or a little light flash. And the idea is that then they can then sort trials into the seen trials when they pushed a button and said, yep, I saw a flash, and the unseen trials when they didn't. And they found that on the seen trials, they were much more likely to have bursting of these layer 5 pyramidal cells and coupling between the apical and the basal compartments. And on the unseen trials, there wasn't. So there's really beautiful empirical work to suggest that, this bursting in these layer 5 pyramidal cells is really important. Brandon's modeling work that I spoke to refines that slightly and says what's really important is that the system is able to burst, but that not the whole system is bursting. So being awake and conscious and aware is really a little bit like a signal to noise boost for a small proportion of your inputs. So it's not that when you wake up and let's say the lateral hypothalamus comes down and wakes up the locus coeruleus and the raphe and the PPN and the basal forebrain and starts to douse the brain in these neurochemicals that then change the gain of the system. You don't want to do it such that all of them turn up that sigma parameter we talked about before to 1, so that now everything's bursting. That's a problem. Yeah. In fact, you're probably likely to get something like epilepsy. Yeah. What you want to do is you want to tune it such that a small subset are bursting and the rest aren't. And here's why that's important. Now that bursting subset can have an influence over whatever comes next. Mm-hmm. If they're bursting, everything you see in the next little moment will be completely defined by the context of that bursting subset. And it turns out that this characteristic, this shape, is something that Dan Dennett spoke of a long time ago. It's what he calls fame in the brain for his model of consciousness. And the idea is that consciousness is kind of like the most famous subset of the brain that is the most active at a point in time. It has a constraint on what comes next, and it has a sort of almost like a sort of idealized status according to its overactivity. And in a way, this kind of like robs consciousness of some of its mystery, but it also explains a lot of what consciousness is, because at the end of the day, consciousness is contextual processing, right? It's the fact that when I'm awake, I will act in a way that I would not have acted if I was not awake. I am grabbing this part of information, I'm letting that information change the way that I will function. And then to bring it back to the basal ganglia will then be incredibly important for changing what it is that becomes famous, right? They're going to have a huge influence over the subset that end up bursting. Where this gets really fascinating and fun is to think about the fact that a lot of models, Bjorn Brems has done some really lovely work here, some really nice models of what we would call free will or sort of volitional agency in the brain. All that they really require is the ability for the system to have a little bit of flexibility that's constrained by the system's architecture. So you don't want it to be that every time you act, the system might do something different. You don't want it to you don't want to be driving your car and all of a sudden the car sort of like jerks off the road. what you'd like is that the car is driving, and based on what you see right now, let's say a gas station versus a fast food outlet, depending on whether or not you need petrol or you're hungry, the system is able to be sensitive to that new input, right? That's really what we need to give the notion of free will some real oomph. And so I'm really excited by the idea that something like the basal ganglia could be really important for kind of imbuing the system with that variability. We call it constrained variability, the variability that is related to what you care about now and can change the way the system acts. Admittedly very speculative, but really fun to think about how these things all link together.
Beautiful. This implies so many things, but and I would have a ton of questions, but maybe to focus a bit more on the specific field of this podcast with the brain simulation. In our field, the cortical basal ganglia, thalamus-cortical loops are all the buzz. They have been since a few decades, and with some exceptions, maybe prominent exceptions. I mean, such as the dimmer switch model of tremor proposed by Helmich around 2010. The cerebellum is often ignored largely, or we seem to have refined maybe the rate models of the basal ganglia over the years. And we speak of parallel loops for motor, premotor, associative, and limbic and all that. And people have also slowly begun to see strong parallels between, the rate models and maybe the reinforcement learning models of the basal ganglia. In these, the basal ganglia act as the main axis or the actor, and then there's the dopaminergic centers that are the critic or the teacher that reinforce actions by pouring dopamine over large territories of the striatum. And we also know that a loss of dopamine leads to a loss of movement. And some claim that in the end, this all can also be nicely explained by reinforcement learning models as well. But how do we now connect these concepts? Maybe the movement disorders clinical, no dopamine means you're bradykinetic, and the reinforcement learning literature and your concept of how would you connect the things, the dots here?
Yeah, great question. And part of me wants to say, please ask me in a few years once I've had a time to think about it a little more. I think you're hitting the nail on the head though, which is that to have utility clinically, there needs to be ways that a model or a theoretical viewpoint moves the needle in terms of what you would have done, right? And I think Rick's work is a really beautiful example of how, embracing, neuroanatomy can help you work out some, novel ways to think about the system, giving rise to a particular set of symptoms like tremor, the cerebellar VIM connections. I think it's like a really compelling way to think about how that kind of a function would occur. Part of my guess for how to link these things with reinforcement learning is that you're going to need to change how reinforcement learning is thought about a little bit. So reinforcement learning is a way of modeling at a kind of real systems level, but with the agent as the kind of modeling exemplar. You're trying to work out if an agent was in scenario A, how do they choose outcome A, B, or C? Are they going to go and look for the reward in the first corridor, the second corridor, the third corridor, depending on what kind of situations you give them. And then all of the modeling is then based on how to work out how to model that agent's behavior. And that's this like N of one explanatum up in the sort of sky. And then we have the neural processes that link to that. Now, one of the parts that makes it so hard to link between those two is that there's no one place in the brain where, function is. It's really distributed. So, we talked before about this idea of system 2 being this kind of like maybe, a hypothesis is that it's the subset of layer 5 pyramidal cells that are burst firing at a particular point in time, maybe contacting the matrix, thalamus, and the colliculus that form the basis of the conscious percept, let's say. That's still an incredibly high dimensional notion, right? Multiple pyramidal cells, each which have their own connections and own characteristics, own firing receptive fields, things like that. And so the question of whether or not our self can be modeled effectively as a one or whether or not you still need to find ways to like link those things together, is a really interesting question. One, I don't have anything super wise to say in that space, but one thing I will say is that there's a lot of really fascinating mathematics around how populations of animals make decisions, like ants deciding when to go out looking for food or when to stay and defend the nest, or bees deciding when to move from a particular nest to another nest. And these are examples of what people would call sort of distributed computation or distributed cognition, where each individual is but one part of a multiverse of different options. And as a whole, they somehow come to a consensus as to how to act. And then you get this emergent behavior out of that system that's really low dimensional. But the rules that govern it are really high dimensional. And that the mathematics of that, for the physicists out there, follows something like a pitchfork bifurcation, which is another type of attractor that we talked about before. And I don't quite know how the answers will look here, but my guess is that if we can follow the neurobiology up and start to ask questions about how a distributed system cells with the constraints that we think the basal ganglia and the cerebellum and the thalamus, etc., impose, could make decisions, does that look like reinforcement learning models? And when does it and when doesn't it? That's the kind of question that I'm excited to ask. I don't have anywhere near enough experience in the reinforcement learning field to be able to come from the other direction and say, here are the two leading theories of reinforcement learning, A or B, and whether we can arbitrate between them. That's why you collaborate, right?
Of course, yeah, makes sense. Okay, so a lot that remains to be studied. And one last thing I wanted to ask more and type of more detailed questions that you mentioned before. You said that you think of the basal ganglia as a dimensionality reduction system, like a sort of funnel coming from the cortex. You go to the striatum, that is already smaller. Then you go to the external pallidum, that's already smaller. Then you go to the internal pallidum, that's even smaller. And there's some STN involved, that's even smaller. So I totally am on board with that. And then you go back to the thalamus and it projects again, maybe more or less to the whole cortex. So you have some sort of the architecture of an autoencoder, or going from high dimensional to low dimensional back to high dimensional space. And just because you mentioned it before, that we could talk about that, what are your thoughts on this, and how crucial is that for the brain?
Yeah, it's a great question. So, I mean, how crucial is it? Well, I mean, Sten Gruen has just shown that the basic sort of architecture of the basal ganglia is pretty much conserved over, something like, was it like 500,000 years or something? So I think it's really important. And I whether it's important for things that we would kind of call quote unquote intelligence, or whether it's important for things that we would call sort of adaptive behavior is another really interesting question. As an organism that has to kind of ultimately decide to do a particular thing. Let's go back to the analogy before of me driving down the road and seeing a petrol station and a fast food outlet. If I both need petrol and I'm hungry, I can't both get petrol and eat. I have to make a decision. I have to like fall off the cliff in one way or the other. If my system was set up such that I could do multiple things at once, I'd probably be organized more along the lines of an octopus or something, or a slime mold, where you can distribute out and achieve multiple things at once. But by being a multicellular organism, a mammal, we are really constrained into particular actions. And one way to think about what the basal ganglia does is that it sort of takes the temperature of the neuromodulatory system, the valuation system, and then imposes its constraint on the subset of layer 5 pyramidal cells that will burst. It like makes it such that if I currently value food over petrol, I will go to the food outlet and get food. Whereas if I currently value petrol over food, I'll go to the petrol station. So in a way, it's sort of like shaping the system such that it has adaptive biological behavior. Now, a fascinating question is whether that's actually a good thing for AI or not. Right? Do we even want our artificial intelligence systems to have that, or is that a constraint that something like AlphaGo doesn't need? Right? If it's going to beat the best players at Go or chess, it's actually an imposition to only do one thing at once. It would be better if the system could try billions of different things and then, come up with the solution. Yeah. So in a way, it's sort of like, it's interesting to think about these things, but it's also really important to remember that, the basal ganglia itself is extremely embedded in the biology. Now, whether the particular bow tie is important, I'd again think, yes, it seems like there's something really important in funneling down and then expanding out. This is something that we see. It's a kind of characteristic feature of complex systems that we see again and again and again, where you'll see this kind of collapsing down of the dimensionality of a signal, only then to reinvigorate the signal. And what's really interesting is that happens in multiple places in the brain, right? So the basal ganglia is one where you see this collapsing, but as the cerebellum receives input from the cerebral cortex, you're right. It's the layer 5 cells send down the projection to the pontine nuclei. There's a many-to-many connection there. The pontine nuclei then project to the granule cells of the cerebellum, which are the most numerous cells in the whole brain, in a many-to-many fashion. So you've got this huge expansion, right? They contact the Purkinje cells' parallel fibers, which then Purkinje cells then project back to a tiny population of deep cerebellar nuclei, which then contact another tiny population of core cells before coming back to the cortex. So there, what you have, instead of having the collapsing and then re-expanding, you've now got the expand, expand, expand collapse. And understanding the benefits and costs of organizing systems like that, I think is actually one of the really, really exciting things that will help to really, open up our understanding of, the sort of architecture of the brain in a language that people like deep neural network people could understand.
Could it be that could one factor be that manipulating data in a low dimensional space is just simpler? For example, it would be harder to form something like a decision on the whole cortex just because it's so distributed. And maybe it's easier to do that in a part of the brain where the information is somehow condensed, and then you have to funnel back up to. And it's not only about decisions, it's about manipulating information, I guess. That could just be easier in the low dimensional spaces to some degree.
I 100% agree. I was actually chatting with some postdocs last week about writing, and we were kind of finding this commonality wherein if you're trying to write something down, a paper or something like that isn't yet clarified in your mind, it's still a bit of a jumble, you don't quite know the structure or whatever, it's really hard to do. And you could spend hours on it typing away sentences and things just don't come out. But if you take the time mentally to kind of organize your thoughts such that you have worked out a structure, and this doesn't have to be entirely mental, it could be writing down a logic of a structure at a really high level. But as soon as you find a way to sort out the system such that it's now low dimensional, it turns out that the writing is much easier and it can really flow out. And so I think there's really something to that, right? That low dimensional descriptions of data are just much more wieldy. They're just easier to handle and juggle and move around. And now you can take the first paragraph and flip it around to the fourth, and the fourth down to the discussion. And those are things that we're capable of doing because they're things that we can keep a hold on. Whereas if you've got 1,000 sentences versus 10 paragraphs, the 1,000 sentences are just going to take you forever. But the 10 paragraphs you can start to mess with. And I think there's really something to that.
Great. So as you see, there would be tons of questions more that I would have, but I have to also value your time. Is there anything you think we missed about the paper? If not, I would have some more general type rapid fire questions to wrap up.
No, I thought your questions were great. I'll just say that if anyone has questions, I'm more than happy to answer any emails or chat to me on Twitter. I'm more than happy to chat.
Amazing. So very quick questions. What research question would you think someone should study, but you don't have the time to do it yourself?
Wow, that's a really, really great question. One of the things that we're really fascinated in at the moment is the kind of logic of how the brain communicates with itself without a controller. And so I think that's an area that I think there are millions of questions, and it doesn't matter if you come from philosophy or neuroscience or engineering. I think that's a really amazing question. What is the kind of language of causal interaction in a complex system with no one region being the CEO? That's the real key question. I'm trying to work on it, but I think the more the merrier. I'd love to have more people thinking about that kind of an idea.
Great. Did you ever make a mistake, Mac?
Yes, almost too many, I'd say, Andy. I think I'm actually a firm believer in the idea that you don't learn anything by just doing what you already know. I think there's this great Kurt Vonnegut quote where he says, "We must always be developing wings on the way down." And I firmly believe in that, and I think it's a really, really important feature for a scientist to have. If you stay in your world and on the thing that you're good at, it's like developing a little silo, and you start to sort of like get into this mindset of defending people from your silo. Don't come near me. My results are perfect, whatever. I have the opposite. I'm like, I think this is all fascinating, and I want to learn about it, and I want to be around really smart people. I have people around me that are far smarter than I am. My collaborators are far smarter than I am. My students are far smarter than I am. I'm trying to bring them together, point to stuff that's really weird and curious and exciting, and just try to be a part of an interesting conversation about how it might work. I think that's a really, really fun way to practice science, and it lets you navigate the grant rejections and the rejections from journals by 4 of your peers that nearly kill you, and see the positive side of it, which is that you get to be professionally curious. And I just love that. Any
eureka moment that you would like to share, or like stroke of insight or success that stuck out?
Yeah. The one with my dad asking me about Tyler learning to walk and then thinking about the fact that the cerebellum was just set up to do it. We didn't talk about it much, but the implication from that is that by standing up and forcing the brain to deal with that extra challenge of this weird kind of heavy head on this really awful bipedal with 2 hips, bipedal legs with unsteady hips, was such that the extent to which the animal could delegate that to the cerebellum and start to anticipate the movements of the hip was a real computational advantage for that animal. And so we started thinking about whether or not the ability to delegate all behavior and to do it in a cognitive fashion would actually be beneficial for humans. And I really do think that that's a really good explanation for what formed the basis of our ability to have this conversation. Language is a beautiful example of a particular set of input-output rules that we automatize. I never think about the sounds that go into a phoneme until I'm trying to teach my 6-year-old how to read, or the verb endings. We just automatize all this stuff so that we can have this fascinating conversation at a high level. Now, my hat's off to you as someone who's speaking in a language that wasn't their natural born language. That's like a whole other layer of complexity. But it's probably even more acute to you that the more that you can automatize English, the more that you can think in high-level concepts in English.
Yes.
And so I really do view the ability to automatize and to delegate behaviors to the cerebral architecture as a really fundamental aspect of our cognitive performance. And I'm really excited to try to unpick and carve some of that stuff out in the next few years.
The paper we discussed is really impressive, as I mentioned, and I had to focus on this one given its depth here. But you've also published amazingly high all the time and seem to pump out all these, high tier papers. The paper we discussed also shows, in my view, that you read a lot. And given that you're maybe generally more the computational modeling person, you seem to also read a lot outside of your own arena, too, as you mentioned a bit as well. Any tips to make all that work? Like, what's your secret, and what to read, how much to read, and then how to be so successful?
Yeah, I'm flattered, Andy. Thank you very much. My wife would say that I read too much. We just recently had to recruit my 2 sons to come help clean up all of the papers that I'd printed out and highlighted all over my office floor. That was one of the other benefits of COVID, is that we had the time to help organize my office. So look, I think there's no secret. I think being curious really helps. I think one thing that is really useful is to think long and hard about what you actually truly find interesting. If you can put that effort in at the start and think, how on earth does this thing work? Let's say you're a clinician and you're really curious as to why some people that get deep brain stimulation with Parkinson's end up being having manic side effects, and other ones can't sleep, and other ones get weird dyskinesias or something like that. If that's the thing that absolutely fascinates you, then now all of a sudden you've got a target for your inquiry, and you can go after that. You can find the papers, and you can really try to break it down. And I'm not saying that causes the whole deck of cards to fall over and the answer presents itself, but I think that's a huge part of it. The other part is something that I'm really a big fan of Richard Feynman and the way he kind of approached being a theoretical physicist. And one of the quotes that he has is that he doesn't understand people that try to learn things by rote. You need a model. You need to have a model of the thing you're trying to understand. And if you do, now all of a sudden it's not just regurgitation, but there's missing parts of the model. And I think one of the things that I've worked really hard to do, and this is going back to finishing medical school and being really dissatisfied with the kinds of stories that we were given about the brain. We had this great story about the heart, a great story about the lungs and the kidneys and the liver, and then they'd get to the brain and people would just sort of throw their hands up. And I was really dissatisfied with that, and I really wanted to kind of make better models and stories of the brain. And so, for the last 15 years, I've been really, iterating and thinking deeply about that problem. And so I'm not saying that this thing kind of like just, click happens overnight, but by having a guiding principle of what you're trying to understand, by developing that model, you essentially make it such that you can it's like the basketball player shooting the shot. You can go and read a paper and inadvertently update your understanding of some other part of the space of the model that you didn't realize. And I think that really, really helps. And so I would encourage everyone to, find the question that really, really kind of, makes the hair on the back of your neck stand up, you get really excited by, and then work really hard to, collapse your misunderstanding and think long and hard about developing your intuition about the system, because I think that really helps. And then you just have to write so much, man. You have to practice writing. You have to really force yourself. The big thing I'll say for any students out there is that, I've published a few papers, but I'd never, ever write it the first draft. The first draft is always awful. It's disgusting. It's gross. I wouldn't show anyone the first draft. I wouldn't even show my wife the first draft of the writing that I do. You have to write it. You have to get it down. You have to get it out of your brain onto the page, and then you can start to shape it and make it nicer and clearer and clean. And the paper that we discussed today, I must have gone through 100 different edits of that paper, if not more, just tweaking it and thinking about how to craft it. And I think you have to really work on that as a skill.
So really spending time to write, also spending time to think, I guess. Do you do that? Like, do you think, next 2 hours I'm going to think? Is that something you do?
I think part of it is trying to be really honest with yourself about the parts of what you know and understand feel strong and the parts that feel flimsy. Yeah. I try to read so I feel like the reading that I do is in like two really clear categories. One category is, oh man, that looks awesome. Like that's a cool paper. I just want to read that. That seems wicked. The other one is there's something I don't understand about that, and I really want to get it to know it better. So, microglia, they're super fascinating. They can release adenosine. Adenosine can then go inside cells, change G proteins, can down-regulate the excitability of cells, but it does it on a timescale that's much slower than neurons. Wow, that's so cool. Like, what kind of an effect does that have on that little sigma parameter we talked about earlier in figure 3? Thinking about the cerebellum and the different kinds of modes of processing of the inner neurons. There's just these areas that I know a little bit about, but nowhere near enough. And I try to really focus in on the areas that I know in my own mind are not clear. Yeah. And I try to be really critical on that. And that's again, really challenging. But fortunately, neuroscience is just chock full of amazing stuff. And so you can really find a lot of really good literature out there to kind of diminish your ignorance about the system.
Do you still see patients?
I do not. Okay. So my PhD, I was on call at one of the local hospitals, and it got to the point where I was having to Google stroke criteria on my phone, and I realized that I just wasn't doing the patients an appropriate justice. So I decided to duck out and really focus on academia. And then after my PhD finished, I got a fellowship, went over to work with Russ in the States, came back. I got another fellowship. I've just got a third one. It's sort of going from, fellowship to fellowship in a way that's sort of insulating me from that world. And I miss large parts of it. The patient contact is something that I held really dear. I didn't leave medicine for a lack of caring or love for the job. It was more that the way that research embraces uncertainty was something that really suited me. Whereas in the clinical world, you really have to, and I think you have to end up coming up with a really certain decision for your patient's benefit. And that was something that just never sat well with me.
You should not be creative, right?
Yeah. It's best if you just know the answer and do it, right? So I found that really suited me. But I still work with a lot of clinicians and collaborate, and I want to get closer back to that space for sure.
So what will you do now that you have explained the brain to conquer the world? What's next is the question.
That's hilarious. So look, I think one of the things I love about science, right, is that it's like another Feynman quote. It doesn't matter how beautiful your theory is, if it doesn't agree with experimental evidence, then it's not right. So having an idea is fun and a great experience, and I've really loved the deep thought that I've gone into with this paper. But the next test is to go out there and test these ideas and see whether or not they hold up. One big challenge there that became really acute to me after finishing this paper was just how big the gulf is between what is actually happening in the brain and the kinds of things that we measure with, let's say, EEG or fMRI or calcium imaging. And so this is what physicists would call an observation model. And so I actually have become really, really interested in trying to work out how does, let's say, activity of layer 5 pyramidal cells versus layer 2 pyramidal cells versus interneurons affect the bold signal versus EEG versus spiking, and trying to think long and hard about how those things work together, because until we have that language sorted or that process clarified, we could have the coolest idea we wanted in the circuitry or the most beautiful pattern we ever saw in the brain imaging, but without being able to link them together with any confidence, we're kind of sort of still in the dark.
That makes sense.
So I still think there's a long way to go.
So last question, how does the future of neuroscience look like?
Yeah, I'm really excited by where we're at with neuroscience. I feel there's a lot of really great data out there. I think if you're a young, enthusiastic scientist with some good questions and a couple skills, there's a lot of really great data sets out there that you can start to kind of analyze and ask really interesting questions on. There's a really strong collaborative effort at the moment across neuroscience too. I really like the idea that different groups are sort of like merging and binding together as needed, depending on expertise. I'm really interested to see what happens post-COVID, assuming we can get on top of this awful virus, that, there's now a little bit less of a constraint on people being, wedded to particular environments, like going into the office. I think scientists, particularly systems neuroscientists, are really well set up for that, where if there's a data set that can be shared and a Zoom call can be made, then we can make progress, I think, a lot more rapidly than we could have in the past. So I'm really excited by the idea of clinicians and neuroscientists coming together with cognitive scientists and try to sort of blend ideas together so that we can start to say, all right, if this is our model of the brain and we think that, this part's broken, what part could we hit in order to make the brain work a bit better? Or what chemical could we add in that would make the brain function in a slightly better regime for a little longer while we fix this other part? So I'm really excited by that. I think that's a huge effort, and it's not going to be the kind of thing that we solve in a year, but it's the kind of idea that we could bring people with different expertise together in the same space to start to have a common conversation about how we can actually intervene on this complex system in a way that's actually going to help in the long run.
Sounds like an amazing future. So Mac, thank you so much for your time in this. It was a great honor to hear you about this incredible paper. And I really would recommend it to most people to read that are interested in the brain. I think it's really tremendous, and it can open up new ways of thinking about the brain. So thank you so much for your time.
Thanks, Andy. It was really fun.